This berry vitality was measured back in 2008. A PhD student of mine used a technique with fluorescein diacetate, or FDA stain, which on a half section berry shows up quite nicely the regions in the berry that are vital. So these are the green staining regions, and the regions which are dead. At about the same time, Krasno et al. From Davis showed the same thing using the same technique. And basically what we found was that the berry maintained vitality up to about 100 days after anthesis. And after that, depending on the cultivar, you see a reduction in that vitality. And this was exactly what they showed as well for Chardonnay, Nebbiolo, and Cabernet. We also examined table grape, and that didn't show any sign of this cell death. So it was peculiar to these wine grapes, didn't occur in table grapes. Subsequently, as shown by Clark et al. Using a different stain, also with Shiraz, that this loss of vitality also correlated with the propensity to split. So because of the breakdown of the membranes in the berry, if they're becoming less vital, it resulted in the berries not splitting, so that their osmotic competence was lost when there was a reduction in cell vitality. This loss of cell vitality was originally proposed by Lang and Thorpe back in the early 90s, late 80s, and was thought to occur at veraison, but these more later experiments showed that it occurred later, usually about 30 or 40 days after veraison. So here's another example of the development of this cell death in berries, where we're plotting the cell weight as a function of the days after flowering or days after anthesis. We've got the vital stains here. In this case, we're using 2 at the same time. So the FDA stain, so all the living cells are green, but also propydium iodide, which stains DNA. And when the cell membranes break down, the propydium iodide can get into the cells and stain the DNA. So we see a stain when the cell membranes break down. And this is just showing you also the shriveling that's occurring later on after this maximum weight's occurred. So generally, we find that this onset of cell death occurs pretty near to the maximum weight of the berry. So you get this in Shiraz around 100 days post-flowering. This is another example of plotting this Shrivel index as a function of living tissue, showing the effect on Shiraz, and also exemplifying the big difference with Chardonnay. So Chardonnay can show a little bit of cell death, but it doesn't show the Shrivel. So we were measuring berry cell vitality with this stain, which is quite laborious and requires a fluorescence microscope and sectioning, etc. And we wanted to come up with a more convenient technique. And so we developed a technique using impedance spectroscopy. And this was done with Luciano Carabia, who did his PhD with me. So we connect some electrodes across the berry, and the system here basically imposes a membrane or potential across the membranes in the berry at different frequencies. And from the response, we can model the cross-section of the berry as electrical components. And using that, we were able to model it with these components, which correspond to a cell pathway and an apoplastic pathway or cell wall pathway. And from the model that we developed, it was possible to very accurately predict the decline in living tissue with time, here plotted as bricks through development. So this is living tissue. The green points are the stain, the vital stain. The black points are the model that we developed from the impedance spectroscopy. Now, this shows that there's another way of measuring it, but it also shows that the stains are not an artifact. In fact, it shows that the cell membranes within the berry are actually becoming leaky. And this is caused by a reduction in capacitance, membrane capacitance, and also an increase in cell wall conductance due to ion leakage from the cells. So after we developed that method, we also wanted to look at the effect of temperature, and Luciano developed some shading protocols and trials over 3 seasons. The industry at that time were interested in shading, and we tried different shading methods. So this was one example using overhead shade, but we also had canopy shade where we had shading across the canopy and also soil shade. It was only the overhead shading that actually had an effect on berry, shrivel, and living tissue. And this just shows you living tissue as a function of days after anthesis. Control, which is the red one is controlled, so unshaded. This is the shaded. The black points are the impedance spectroscopy measurements. The green and red points are the vital stain. And you can see that the shade results in a prevention of that loss in cell vitality, and that was also evident in the shrinkage. We also noted, however, that water stress had an additional effect. So in addition to the temperature, and here we're plotting it as thermal time, living tissue as a function of day degrees, you can see that the values for the treatments line up on the thermal time, but with increased water stress, it tended to move it down. So it probably indicates that water stress is having an additional effect. To follow that up, Zhayu Zhao did a trial for his PhD where we used the Victor Sadrason saw technique for heating vines. So this is a series of panels that allow solar radiation to come in around the vines, and then through convection, heat rises up through the canopy and increases the temperature within the canopy and the bunchstone. So using this technique, you can get elevated temperatures within the vine. So this occurred in this trial between 0.5 and 1 degree centigrade. So we had a combination of treatments where we had heated and irrigated and heated and non-irrigated. So the non-irrigated vines experienced some water stress. In fact, there was a 50% reduction or more in stomatal conductance. When we look at living tissue as a function of time, thermal time in this case, we see that most of the temperature treatments lined up together. So that illustrates the importance of temperature, but with the non-irrigated treatment, it shifted it across, indicating that water stress had an additional effect.